So I am starting today tetracycline. So first of all, we are discussing the historical background of the tetracycline. You see here, actually tetracycline is a broad spectrum antibiotic. Actually, it is a large family of broad spectrum antibiotic, which were discovered by Benjamin Menji Dogger in 1948. You see here. So tetracyclines are the large family of the broad spectrum antibiotics which were discovered by Benjamin Menji Dogger in 1948 as a natural products, or it is first prescribed in 1948. It is first prescribed in 1948. Benjamin Dogger, working under the Y. Subbarao, one scientist who was working in the Lateral laboratories, discovered the first tetracycline antibiotic, that is chlortetracycline. That is the first discovered tetracycline is chlortetracycline, which is also called as aureomycin. Remember, which is also called as aureomycin. In 1945, it is isolated from the bacteria Streptomyces aureofaciens. It is obtained from the Streptomyces aureofaciens. That's why the antibiotic also designated as aureomycin. So first tetracycline was discovered by scientist Duggar in 1948. In 1948, and it is obtained from the bacteria Streptomyces aureofaciens. That's why the chlortetracycline also designated as aureomycin. Aureomycin. Next, subsequently in 1950, the scientist R.V. Woodward, who was working in the Pfizer, discovered the second tetracycline, that is called oxytetracycline. It is also known as terramycin. It is also known as terramycin, and it is obtained from the Streptomyces rhamnosus. Streptomyces rhamnosus. So semi-synthetic tetracycline obtained from the chlortetracycline in the year of 1953. In the year of 1953. See now, in case of historical background, first the tetracycline was discovered by Duggar in 1948. So in 1948, the first tetracycline was discovered. The name is called as chlortetracycline. Chlortetracycline. This chlortetracycline is obtained from the bacteria Streptomyces aureofaciens. That's why, on the basis of the source of origin, that chlortetracycline also designated as aureomycin. Subsequently, in 1950, scientist R.B. Woodward also discovered the second tetracycline, that is oxytetracycline, which is obtained from the Streptomyces rhamnosus. Later on, 1953, all the congeneric derivatives or semi-synthetic tetracycline was discovered from chlortetracycline. So this is the brief historical background of the tetracycline. Next, go for chemistry and stereochemistry of the tetracycline. Chemistry and stereochemistry of the tetracycline. You see here, I draw the structure. You see the structure of the tetracycline here I draw. Basically, this tetracycline basically contain, what does it mean? Tetracycline contain how many rings? 4 rings. 4 cyclic ring contain. So here what, based on the above conventional structure, you see the various compounds are formed by the making the substitution. See here, this is the structure of tetracycline. You see here, in one position ketone group and 2 is the carboxamide functional group. Here this is the 4 ring A, B, C and D, and these are the numbering here giving. So here the various semi-synthetic tetracycline is obtained by the substitution at position C5, C6, C7. Now presently the name tetracycline refers to a number of antibiotics of either natural or semi-synthetic origin derived from the system. Means whatever tetracycline is obtained, that tetracycline is either semi-synthetic or natural. Both semi-synthetic or natural tetracycline consist of linearly annihilated six-membered ring, that is 1, 4, 4a, 5, 5a, 6, 11, 12a octahydronaphthacene. Means whatever the already I told, the semi-synthetic tetracycline are obtained from the chlortetracycline in 1953. So whatever new semi-synthetic derivatives obtained from the chlortetracycline, all of the tetracycline contain four linearly annihilated six-membered rings. Four linearly annihilated six-membered ring, which consist of 1, 4, 4a, 5, 5a, 6, 11, 12 octahydronaphthacene. The basic nucleus is called octahydronaphthacene. Now, the tetracycline molecule possesses a 5 asymmetric center. It consists of 5 asymmetric center. You see here, the asymmetric carbon atom are present, that is C4, C4a, 5a, C6, and C12a. So these are the asymmetric carbon atom present in the tetracycline. So this is the basic structure for the tetracycline, what I have shown. Now we are going to discuss the chemical properties of tetracycline. Chemical properties of tetracycline. You see here, first it has two properties. One is called acidity constant. Second one is called chemical properties. Now, before going to discuss this one, I am explaining here. Chemical properties, here two properties we should discuss. The major properties, number one is called acidity constant of the tetracycline. Second one is called chemical reaction of the tetracycline. The tetracyclines are congeneric derivatives of the polycyclic naphthacene carboxamide. Basically the basic ring of the tetracycline is called polycyclic naphthacene carboxamide. If you consider these two rings, if you consider this A and B ring, if two benzene ring is attached together, that is called naphthalene. So here two ring is there remaining. See, these are the cyclic ring, and this cyclic ring attached with another ring. So this we consider the naphthalene. So we can basically tell that the tetracycline consists of polycyclic naphthacene carboxamide. Basic nucleus. See here we are discussing the acidity constant. So before going to the acidity constant, basically tetracycline consists of three acidity constant, that is pK1, pK2, pK3. Now these three acidity constant, why the tetracycline shows these three acidity constant, for which functional groups are responsible for that. So I am here discussing, you see here. Now you tell me, anybody, what is acidity constant? What does it mean, acidity constant? Anybody? Hello? Hello? Anybody, acidity constant, what does it mean, acidity constant? Anybody? Abhay Kumar Gupta?
Yes, sir.
Acidity constant, what does it mean, acidity constant?
The acidity constant मतलब ionization constant.
हाँ, ionization constant. तुम define करो, what does it mean? What is acidity constant?
एसिडिटी मतलब आयनाइजेशन कांस्टेंट मतलब ये है कि मतलब कितना पीके वैल्यू है, ऐसे मतलब कितनी लिपिड सॉल्युबल ड्रग है, कितनी मतलब वाटर सॉल्युबल ड्रग है. आयनाइजेशन का तो मतलब यही होता है. अगर वो लिपिड सॉल्युबल.
ओके, डिफाइन करो, व्हाट डज इट मीन एसिडिटी कांस्टेंट? यू डिफाइन इट. व्हाट इज एसिडिटी कांस्टेंट?
डेफिनेशन.
डेफिनेशन, यस. This is very important for you, tetracycline. It will come maybe 2 marks or 3 marks. Okay. So acidity constant means this acidity constant we can apply for which type of acid or base? Tell me.
weak acid, weak base.
Now, what are the name of that compounds? For example, acidity constant is also known as another name, ionization constant. So another name,
Yes sir, but
another name, ionization constant.
particle coefficient
Huh? No, partition coefficient is different.
नहीं, वो तो
Acidity,
डिफरेंशिएट वो तो अलग है।
हाँ, एसिडिटी कांस्टेंट का दूसरा नाम क्या है? आयनाइजेशन कांस्टेंट। और भी एक नाम है, क्या है उसका? फिजिकोकेमिकल
The
प्रोपर्टी।
dissociation,
Dissolution
अच्छा,
constant.
हाँ, वेरी गुड।
The dissolution constant.
Dissociation constant, not dissolution. Dissociation constant, exactly. Dissociation constant. Now, what does it mean? Actually, dissociation constant or acidity constant or ionization constant, we can apply it for the conjugated acid or base. Conjugated acid or base. So, can you tell me what does it mean conjugated acid or base? Can you give example of conjugated acid? Already you have studied in the physiochemical parameters. It is basic question. What does it mean conjugated acid and base? We can apply the pK value for conjugated ionization of the conjugated acid or bases. No idea. Already MC1 passed out. What conjugated acid or base? I won't tell. Anyway. So what does the acidity constant mean? Acidity constant is defined as a measure of the strength of an acid or a base and is denoted by pKa. Remember, the acidity constant is a measure of the strength of an acid or a base, which are applied for the conjugated acid or bases, and it is designated by pKa. It is designated by pKa, and it is also designated as ionization constant or dissociation constant. Ionization constant or dissociation constant, as the substances are able to ionize or dissociate depending upon the solvent. Now, the tetracycline molecules, it exists three acidity constant depending upon the three types of functional group present in the molecule. So first of all, we are discussing, you see, the pKa, pKa1, pKa2, pKa3. See here. So tetracycline molecule consists of three acidity constant. So why it shows three acidity constant? Due to the presence of different functional group. First we are discussing is pKa1. See here, this one. pKa1. pKa1 means this is a 2.8 to 3.3. So pKa1, why it is shows? This pKa value ranges between 2.8 to 3.3. This is basically responsible. This pK value is derived due to the conjugated trione system, conjugated trione system between the C1 and C3. You see, C1 and C3. C1 and C3. Here C1 double bond, here two double, here 1 2, here another double bond is there. So this is basically mainly due to the C1 to C3. Means here you see this one C1, C2, C3. That is trione system. Due to this trione system, this will give the pK value 2.8 to 3.3. So first pK value of tetracycline is due to the presence of trione system from C1 to C3. See, this is the trione system. Here I am saying C1 to C3. So due to this double bond, the pK value is 2.8 to 3.3. Next, pKa2. I designated here pKa2. The pKa2 is basically responsible for the conjugated phenolic anion system. Conjugated phenolic anion system between the C10 to C12. You see, C10, C12, C12, and here middle C11. That is C10 to C12. So in the middle, what is coming? C10 to C12 means C10, C11, C12. Means this is basically, that is phenolic anion system. That is conjugated phenolic anion system between the C10 to C12 it will give the pKa2 value 7.2 to 7.8. You see, phenolic anion system C10 to C12. What does it mean? In C10 position, hydroxyl group is there, and C12 position, hydroxyl group is there, and C11 position, ketone group is there. So these two hydroxyl group act as a phenolic hydroxyl group, whereas this is a ketone group. That's why it is called phenolic anion system. Phenolic anion system between the C10 to C12. So the pKa2 value is derived from the phenolic anion system between the C10 to C12. This is the second pK value for the tetracycline. Next, third pK value of tetracycline is 9.1 to 9.7. This pK value is mainly due to the presence of the dimethyl amino functional group of the C4 position. Dimethyl amino functional group of the C4 position. So these are the, that is, tetracycline shows three acidity constant due to the three different conjugating system. Three different conjugating system. The first pK value is derived due to the presence of the conjugated trione system. Conjugated trione system from C1 to C3. Next, pK2 value 7.2 to 7.8 is derived due to the phenolic anion system. Phenolic anion system between the C10 to C12. Whereas the pK3 9.1 to 9.7, it is mainly due to the presence of the dimethyl amino functional group. Dimethyl amino functional group. So these are the three acidity constant of tetracycline due to the three different conjugating system present in the tetracycline molecule. So these are the different acidity constant for the tetracycline. Next, go for the chemical properties. This is chemical reaction. Second one.
सर
हाँ बोलो.
सर मतलब मैं जो एसिडिटी कांस्टेंट आपने बताया वो सही से समझ नहीं पाया, आपने क्या बताया?
कौन सा?
आई एम नॉट अंडरस्टैंड जी एसिडिटी कांस्टेंट, जिसे आयोनाइजेशन कांस्टेंट बोलते हैं.
हाँ, ये टेट्रासाइक्लिन में तीन एसिडिटी कांस्टेंट शो कर रहा है, क्योंकि
ये तो समझ में आया सर.
हाँ.
सर इसके डेफिनेशन जो आपने बताया वो बताओ, मतलब वो नहीं समझ में आया, क्या बताया आपने?
अच्छा, वहाँ पे एक्चुअली जो पी के वैल्यू होता है, पी के वैल्यू इज मेनली हम लोग डिफाइन कर सकते हैं दैट इट इज द स्ट्रेंथ ऑफ एनी acid or base, means how much amount of the it will produce the hydrogen ion or hydroxyl ion. Okay, it is the strength of a acid or base. That pK is the pK is related with the hydrogen ion and OH minus ion. It is both are related with the negative logarithm value of the hydrogen ion as well as the hydroxyl ion. So it is mainly denoted the strength of an acid or strength of an base in case of conjugated an acid base. For example, I am giving for sulfuric acid. Sulfuric acid, it is a conjugated acid. Okay, conjugated acid. So can you tell me in sulfuric acid, conjugated acid contain conjugated base? So here, which part is acidic part and which part is basic part in case of sulfuric acid?
Sir, H2SO3 sir is a base part and H
H2SO4. H2SO4, हाँ. Which one is acid part?
सर मैंने दोनों को मतलब डिवाइड करके बताया है.
हाँ,
H+ होगा वो तो एसिड पास होगा,
हाँ.
और जो H2 और SO3 होगा वो होगा आपका बेस पार्ट.
हाँ, तो H2SO4, if you hydrogen ion, it will give the hydrogen ion and sulfate ion. So here sulfate 10 मिनट बाकी है, you have 10 मिनट बाकी है, फिर वो चला जाए, ठीक है. So sulfate is act as a base, as because already we know base according to Arrhenius theory or Bronsted theory. So base is a base having the negative charge.
OHI.
which was electron extra, they are accepting the electron, so base. Yes, sulfate
Okay.
anion act as a base, while hydrogen ion it acts as the acidic part. So now sulfuric acid is a conjugated acid. It will undergo 3-step ionization: primary, secondary, and tertiary. Remember, primary, secondary, tertiary. So for that, its acidity constant will be different. समझ में आ रहा है?
Yes sir, आ रहा है.
So sulfuric acid undergo tertiary ionization. It will undergo primary ionization, secondary ionization, tertiary ionization. So each ionization it has the different acidity constant. So here sulfate act as a conjugated base part. ठीक है? तो हम लोग जो भी पी के वैल्यू कंसीडर करूंगा, स्ट्रेंथ we can consider strength of acid or strength of base. That pK is derived by Henderson-Hasselbalch equation, which already studied in the buffers. Once
Yes
you
sir, buffer equation.
buffer equation, once you can deduce the equation, that you can understand. Okay, so now that according to this, that is tetracycline consists of 3 acidity constant depending upon its functional group present.
Yes sir, आ गया, ये तो समझ में आ गया.
Okay. Next, chemical reaction. Actually, tetracycline undergo different types of reaction depending upon the condition or is environment. So first we can see here, in acidic condition, when you expose the tetracycline to dilute acid condition, tetracycline undergoes dehydration to yield a anhydrotetracycline. Anhydrotetracycline, you see here, anhydrotetracycline. If you expose the tetracycline on a dilute acid solution, then it undergo dehydration to give the anhydrotetracycline. That anhydrotetracycline or anhydroteramycin suffers further cleaved and lactonization to give the apoteramycin. This is the structure of apoteramycin. See, this is the structure of apoteramycin. Here I am not showing the intermediate anhydrotetracycline. I am directly showing the apoteramycin or apotetracycline. You see, if you expose the tetracycline to a dilute solution, it undergo dehydration to give anhydrotetracycline. That anhydrotetracycline or anhydroteramycin further cleaved or further undergo lactonization to give the apoteramycin or एपी एपोटेरामाइसिन, इसका दूसरा नाम है एपी टेट्रासाइक्लिन. See, this undergo this is here, this undergo the lactonization. So that's why in dilute solution tetracycline undergo fast dehydration. So it will remove the water molecules by breaking the ring. Then it will form the anhydrotetracycline. This anhydrotetracycline further cleaved and undergo lactonization to give the apoteramycin or epitetracycline. So this is the structure of apoteramycin or epitetracycline. Diluted acid promotes the epimerization at C4 position. You see. Sometimes dilute acid promote the epimerization at C4 position also. See, in that where the dimethylamine functional group is there, sometimes it will also break. Then it will go the epimer. It will go the epimer of the tetracycline. This is the reaction under acidic condition. Next, for basic condition, in mild alkali attacks the carbon of tetracycline, which transform to isotetracycline. You see here, this is the structure of isotetracycline. Means in mild alkali solution, the tetracycline undergo intermolecular rearrangement to form the isotetracycline. This is the structure of isotetracycline. So in case of acidic condition, first of all it will undergo dehydration to form anhydrotetracycline or epiteramycin. Subsequently, that epiteramycin undergo clip and electronization to form the lactone form or epimer. And in the mild alkali solution, it will form the isotetracycline. This is the structure of the isotetracycline. Next, formation of the complexes is a very important point. Remember this, formation of the complexes. Tetracycline possesses a great tendency to form a complex with a number of the chemical species due to its B and C ring oxygen atoms. See here, B ring, C ring. Here oxygen atom is there. Here this oxygen in the form of the ketone, and here this oxygen present in the form of the hydroxyl group. So due to this, it has a tendency to form a complex because oxygen or OH, they are electronegative atom, they can form a coordinate covalent bond or covalent bond. See here, what are the chemical species to form the complex? See, I here written tetracyclic complex most readily with the iron, either ferric, ferrous, cuprous, nickel, cobalt, zinc, manganese, magnesium, calcium, beryllium, aluminium. These all are the metals can form the complex with the tetracycline. And other ions such as phosphate, citrate, salicylates, para-hydroxybenzoates, saccharin anion, caffeine, urea, thiourea, then polyvinyl pyrrolidone, serum albumin, lipoproteins, globulins, and RNA molecules. These all are the chemical species which can readily form the complex with the tetracycline. Remember, this is a very, very important point for the drug interaction as well as the absorption of the tetracycline from the GIT. Tetracycline basically it has the greater tendency to form a complex with the metals or other chemical species due to the oxygen atom present in the B and C ring. So it will form the complexes with the metals, iron, ferric or ferrous, cuprous, nickel, cobalt, zinc, manganese, magnesium, calcium, barium, aluminium, and other ions such as phosphate, citrate, salicylates, ferrohydroxybenzoate, saccharin anion, caffeine, urea, thiourea, polyvinyl pyrrolidone, serum albumin, and lipoproteins, globulins, and RNA molecules. So these all are the chemical species which are readily to form a complex with the tetracycline. So these are the basic three chemical reaction happened within the tetracycline. So tetracycline undergo chemical reaction under mild acidic condition, under mild alkali condition, and with the metal or other chemical species. Next, go for the antimicrobial profile of the tetracycline. Antimicrobial profile of the tetracycline. So already I told tetracyclines are highly effective against a wide range of gram-positive and gram-negative bacteria, such as Rickettsia, spirochete, and large viruses. Means antibiotic basically not active against the viruses, but it has been shown that tetracycline are effective some of the viruses such as members of lymphogranuloma group. Those viruses form the lymphogranuloma. Lymphogranuloma is one type of cancer of lymphoid tissue. So that virus can be killed by the viral replication will be inhibited by the tetracycline. But it does not kill that virus, but they can inhibit the growth and development for that viruses. Those form the lymphogranuloma group. Now, the main indication for the use of tetracyclines, it is basically used for the Escherichia coli, Haemophilus influenza